Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, right, so we're currently in the process of discussing IP3 receptors, okay, and of course to activate IP3 receptor signaling, to activate the release of calcium from IP3 receptors in the endoplasmic reticulum, you need to generate IP3, and we've now discussed how you generate IP3. It is from the hydrolysis of the membrane phospholipid phosphatidylinositol 4,5-bisphosphate, which for short is abbreviated down to PI45P2. Okay, and this hydrolysis reaction here of PI45P2 to diacylglycerol and IP3, the two products, is carried out by phospholipase C enzymes. Okay, so now what I want to discuss is how phospholipase C enzymes can be activated by cell surface receptors. Okay, now, the first thing to say is that there is not just one phospholipase C enzyme. Okay, there are a whole bunch of phospholipase C enzymes. I believe the number is 13 different phospholipase C enzymes, and these are grouped into different families. Now, there are only two families of phospholipase C enzymes that are going to be of interest to us, and we're going to discuss in this video. Okay, there are 4 other families besides these two main ones, giving a total of 6 total families. And if you're interested in some of the more niche ones, I have a video on phospholipase C enzymes which covers all 6 families of phospholipase C enzymes and how they can be regulated. Okay, but we're going to talk about the mainstream ones, okay, which are the phospholipase C beta family of phospholipase C enzymes, okay, and the phospholipase C gamma family of phospholipase C enzymes. Okay, so each of these is a family, so it contains multiple phospholipase C enzymes. Okay, so let me tell you the names of the members of these families then. So the phospholipase C beta family first. This contains 4 phospholipase C enzymes. Okay, phospholipase C beta 1, phospholipase C beta 2, phospholipase C beta 3, and phospholipase C beta 4, and they are all coded for by different genes. Okay, so here we have 4 different phospholipase C enzymes: phospholipase C beta 1, beta 2, beta 3, and beta 4. Okay, similarly, the phospholipase C gamma family has multiple members, okay, but not quite as many. It has 2 separate members, phospholipase C gamma 1 and phospholipase C gamma 2. Okay, and again, they're coded for by different genes. Okay, right. Now, all of these enzymes, all of them, all catalyze this same reaction. Okay, they catalyze the breakdown of PI45P2 into diacylglycerol and IP3, this crucial molecule that we're trying to create. Okay, however, these 2 different families, the phospholipase C betas and the phospholipase C gammas, are regulated, i.e. activated by very different things. So what activates the phospholipase C betas? Well, the phospholipase C betas are the most famous ones. They're the most mainstream ones. Okay, if someone just puts phospholipase C and then doesn't tell you which family they're talking about, you can assume they're talking about the phospholipase C beta family. These are the ones which are activated by G-alpha Q/11 subunits of heterotrimeric G proteins. Okay, so they are activated downstream of G protein coupled receptor pathways. Okay, and we will come on to discussing those G protein coupled receptor pathways in just a moment. Okay. Meanwhile, the phospholipase C gamma enzymes, these are activated by receptor tyrosine kinases, okay, such as, for instance, the epidermal growth factor receptor is an archetypal example of a receptor tyrosine kinase, and I'll abbreviate receptor tyrosine kinase down to RTK here. R for receptor, T for tyrosine, and then K for kinase. Okay, and again, we will look at this pathway by which receptor tyrosine kinases can activate phospholipase C gamma enzymes. Okay, right. So the first thing then to talk about is the phospholipase C beta enzymes and how they can be activated by the GQ/11 cascade. So let's turn over the page and talk about G protein coupled receptors then. Okay, so G protein coupled receptors, usually abbreviated down to GPCRs, are a massive great family of receptors in humans. Okay, so in humans alone, we have over 800 different G protein coupled receptors. Okay, and they'll all have different ligands, basically. Okay, now, they're not just found in humans, they're found in all sorts of different parts of life, basically. And I think the total number of different G protein coupled receptors that we've now found in all the different species that we've studied is now up to 30,000. So we've found a lot of these things, basically. Okay, but coming back to humans then. So G protein coupled receptors are receptors that are in the cell membrane and which are going to respond to extracellular ligands. They're going to bind to extracellular ligands and then they're going to activate intracellular signaling cascades, okay, via heterotrimeric G proteins. So let's just go over this then. Okay, so we'll start off with the famous structure of G protein coupled receptors. So what characteristic then do you have to be considered a G protein coupled receptor? Okay, well, the characteristic feature that all G protein coupled receptors have is that they are a single protein which spans the membrane 7 times. Okay, so they have their N-terminus extracellularly. They then span the membrane 7 times, and they finish up with their C-terminus intracellularly. Okay, so this is the extracellular fluid side of this membrane here. Okay, and this is the cytoplasmic side of the membrane here. So this is the cytosol. So this is the extracellular fluid, this is the cytosol, and this is our G protein coupled receptor. And the key feature that it has here is the 7 membrane spanning alpha helices. Okay, right. So when a ligand binds to a G protein coupled receptor, and there are a huge number of different G protein coupled receptors which all have different ligands which can bind to them and activate them. Okay, but the general principle that unites all of them is that when their respective ligands bind to them, it causes a conformational change in the G protein coupled receptor that is particularly prominent in the intracellular portions. So in these 3 loops here on the intracellular aspect and also on the intracellular portions of the membrane spanning alpha helices. So you get this conformational change on the intracellular aspect of the G protein coupled receptor when the extracellular ligand binds to it on the extracellular side. Okay, and this now allows the G protein coupled receptor to interact with heterotrimeric G proteins. Okay, so let me now talk about heterotrimeric G proteins. I'll extend the membrane so that I can do this. Okay, so heterotrimeric G proteins, the name of these things tells you a lot about what they are. Okay, so heterotrimeric, that tells us that these things are going to be made out of 3 separate subunits, okay? That's what trimeric means. The hetero tells us that the 3 separate subunits of the heterotrimeric G protein are all going to be different. Okay, so let me now draw the 3 different subunits of the heterotrimeric G protein. So they are made out of 3 different subunits which are called the alpha subunit, which I'm drawing now. Okay, so this is representing our alpha subunit. And then they also have a beta subunit and a gamma subunit. Okay, so here's the beta subunit, and here is the gamma subunit. Okay, and I'll colour these in different colours. So we'll have the alpha subunit here represented in red. Okay, so here's our alpha subunit of the heterotrimeric G protein. Then we'll have the beta subunit here in blue. Okay. And finally, we'll have the gamma subunit of the heterotrimeric G protein in green here. Okay, right. Now, heterotrimeric G proteins then can be in 2 states. Okay, and this is the absolutely key concept here. They can be in 2 states. They can be in an off state, okay, and they can also be in an on state. Now, let me firstly describe the off state, because it's the off state that I've initially drawn it in, and indeed, before the G protein coupled receptor has been activated, and before the G protein coupled receptor activates the heterotrimeric G protein will be in the off state. Okay, so what is the features of the heterotrimeric G protein when it's in the off state? Well, basically the alpha subunit has guanosine diphosphate bound to it in the off state rather. Okay, so at the moment it's in the off state, and therefore in this little binding site that I've drawn here, it will have guanosine diphosphate bound to it. Okay, a molecule of GDP here, guanosine diphosphate. Okay, right. Now when the alpha subunit has guanosine diphosphate bound to it, it is in a conformation which allows it to bind to the beta and gamma subunits. Okay, so the entire thing therefore is at the moment as a trimer. Okay, right. Now the beta and the gamma subunits never ever separate. Okay, they are always bound together, so they are often referred to as the beta gamma complex. So we can say that when the heterotrimeric G protein is in the off state, the alpha subunit has GDP bound and it will associate with the beta gamma complex here. Okay, right. Now contrast that to the on state of the heterotrimeric G protein. In the on state, the alpha subunit has GTP bound to it. Okay, and when it has GTP bound to it, it no longer wants to associate with the beta gamma complex. Okay, the GTP changes the conformation of the alpha subunit so that now it can't bind to the beta gamma complex anymore. This interface is broken, and now the beta gamma complex separates away from the alpha subunit, but the beta and the gamma subunit stay bound together always. Okay, so in the on state, alpha goes off on its own with GTP bound to it, and the beta gamma complex goes off on its own. Okay, now the other thing to mention is that the subunits of heterotrimeric G proteins, they are not integral membrane proteins. However, they are attached to the underside of the phospholipid bilayer, as I've shown here. Okay, and the way that they are attached to the underside of the phospholipid bilayer is through lipid moieties. So alpha subunits of heterotrimeric G proteins have a lipid moiety attached, and gamma subunits of heterotrimeric G proteins have a lipid moiety attached. Okay, so these are little sticks that are implanting into the inner leaflet of the phospholipid bilayer. These are lipid moieties. Okay, now for those of you who want to know, this one here is either a palmitoyl group or a myristoyl group. Okay, and this one here is either a farnesyl group or a geranyl group. Okay, now those details do not matter. The important point is the concept here, and the concept is this: that onto the side of these proteins, the alpha subunit and the gamma subunit, you're going to stick this molecule that is going to stick off the side of them, okay, and it's going to be really hydrophobic. It's not going to be soluble at all, okay, so it's not going to interact well with water at all. So what ends up happening is this lipid moiety ends up sticking itself into somewhere else that is hydrophobic, which is the phospholipid bilayer. So it ends up implanting into the phospholipid bilayer, and then the protein ends up dangling underneath the phospholipid bilayer. So adding lipid moieties on like this is a way of attaching proteins to the underside of the phospholipid bilayer. And indeed, the alpha subunit and the gamma subunit are attached to the underside of the phospholipid bilayer by lipid moieties. Now the beta subunit doesn't have a lipid moiety of its own. Instead, what happens is the beta subunit is attached to the gamma subunit and thereby remains attached to the underside of the phospholipid bilayer, since the gamma subunit is attached to the underside of the phospholipid bilayer. Okay, right. The next thing to say about heterotrimeric G proteins is to talk about the diversity of heterotrimeric G proteins. Okay, so there is not just one alpha subunit, there is not just one beta subunit, and there is not just one gamma subunit. There are a huge number of different alpha subunits of heterotrimeric G proteins. There are 16 known genes for alpha subunits. Okay, so 16 alpha subunit genes. Okay, there are 5 beta genes. Okay, so 5 different genes for beta subunits of heterotrimeric G proteins, and 12 different genes for the gamma subunits of heterotrimeric G proteins. Okay, so that means that there are many different alpha, beta, and gamma subunits, because often many of these genes have multiple splice variants, which further adds to the diversity. So you can build a lot of different heterotrimeric G proteins, basically. Okay. Now, coming on to the activation of heterotrimeric G proteins. So basically, when a heterotrimeric G protein is in the off state, as I've drawn here, it is not signaling at all really. It's not doing anything. Okay, it won't be affecting any other signaling molecules. However, they can be turned into the on state where they do activate downstream signaling molecules. And how can they be turned into the on state? Well, by G protein coupled receptors. So when a ligand binds to a G protein coupled receptor and activates the G protein coupled receptor, the G protein coupled receptor's intracellular aspect is now capable of binding to certain heterotrimeric G proteins. Okay? Now, it will not activate every different type of heterotrimeric G protein. Each G protein coupled receptor has its favourite types of heterotrimeric G proteins. Okay? Now, as a simplification, everyone always talks about the fact that G protein coupled receptors select the heterotrimeric G proteins on the basis of which alpha subunit you have. It's not, strictly speaking, true. In reality, the G protein coupled receptor binds to the alpha, the beta, and the gamma subunits of the heterotrimeric G protein, and therefore does select to some degree what beta and gamma subunit you have as well. However, the main subunit that it binds to is the alpha subunit, and therefore the alpha subunit is the main one which determines whether a certain G protein coupled receptor can activate this heterotrimeric G protein. So most people do not make a fuss over what beta subunit and what gamma subunit you have in your heterotrimeric G protein, and they just dwell on the alpha subunit that is present in the heterotrimeric G protein. Okay, so often we talk about how a certain G protein coupled receptor will activate heterotrimeric G proteins with a certain alpha subunit, and we don't faff about the beta and the gamma subunit. Okay, in reality, the beta and the gamma do determine the specificity, okay, but only mildly compared to the alpha subunit. Okay, so now let me tell you about the heterotrimeric G proteins then that we are specifically going to be interested in. Okay, and the heterotrimeric G proteins that we're specifically going to be interested in. Well, actually, I don't think I actually finished the activation story. Okay, so G protein coupled receptors combined to heterotrimeric G proteins. Okay, and then what they're going to do is they're going to catalyze the release of the guanosine diphosphate from the alpha subunit of the heterotrimeric G protein. Okay, and then what will happen is a GTP molecule will come in and replace that GDP molecule. Okay, so normally when you've got a heterotrimeric G protein that isn't bound to a G protein coupled receptor, that GDP is very powerfully bound to the alpha subunit. It cannot just leave the alpha subunit. Okay, however, once the heterotrimeric G protein has bound to the G protein coupled receptor, then the G protein coupled receptor catalyzes the release of the guanosine diphosphate. Okay, it makes the bond between the alpha subunit and the GDP much weaker. Okay, so the GDP is released. A GTP molecule from the cytoplasm will then come into this binding site and replace the GDP. Okay, then the alpha subunit now has GTP bound to it, so it changes conformation. Okay, and in this new conformation, it can no longer bind to the beta-gamma complex, so it splits apart from the beta-gamma complex, and both the alpha subunit with GTP bound and the beta-gamma complex split away from the G protein coupled receptor. Okay, so the overall thing then that you end up with is like this. Here is the cell membrane, and here now is our alpha subunit of the heterotrimeric G protein, like so. So here's our alpha subunit with GTP bound, like so. And I'll colour this in red. And then you also have the beta-gamma complex, which will go off also, like so. So they'll be separated now. Okay, but both of them will still be attached to the underside of the phospholipid bilayer because they both have those lipid moieties attached. So I'll colour in the lipid moieties here in yellow. Okay, so here's the beta subunit now in blue, and here's the gamma subunit in green. So basically, the G protein coupled receptor catalyzes the conversion of the off-state heterotrimeric G protein to the on-state heterotrimeric G protein, like so. And now both of these molecules, the alpha subunit with GTP bound and the beta-gamma complex, can go off and have signaling roles. Okay, now, again, this is another reason why we don't bother that much about the beta and the gamma subunit and which specific one of these they actually are, okay, because it doesn't seem to matter what beta and what gamma subunit you specifically have in your beta-gamma complex. It doesn't seem to affect what actual signaling roles they go on to have, okay? Beta-gamma complexes seem to do the same thing no matter what beta and what gamma subunits you have. So that's another reason that people don't generally fuss about which beta and which gamma subunit you have in your heterotrimeric G protein. Whereas, which alpha subunit you have hugely affects what the signaling properties of that alpha subunit is. Okay, so that's the activation then of heterotrimeric G proteins by G protein coupled receptors. Okay, now we are going to be interested in heterotrimeric G proteins of a certain type, okay, which means that they have a certain alpha subunit, okay. So, G proteins, heterotrimeric G proteins, are named according to which alpha subunit they have, okay, and we are going to be interested in GQ/11 heterotrimeric G proteins, okay. Now, GQ/11 heterotrimeric G proteins are heterotrimeric G proteins where the alpha subunit here is either the very famous alpha Q subunit or it's the less famous alpha 11 subunit. Okay, now these are almost identical to one another and they have pretty much identical signaling properties. Okay, so when we put GQ/11, we mean a heterotrimeric G protein where the alpha subunit is either alpha Q or it's alpha 11. Okay, so I'll put alpha Q/11 here. Okay, and that means an alpha subunit that's either alpha Q or it's alpha 11. Okay, and as I say, we don't care what the beta and the gamma subunits are. It doesn't make that much difference about which G protein coupled receptors can actually activate the heterotrimeric G protein, and it doesn't really make that much difference to the actual signaling roles of the on-state heterotrimeric G protein. Okay, right, so different G protein coupled receptors, as I said earlier, will activate different types of heterotrimeric G proteins, okay? And different G protein coupled receptors will prefer to activate heterotrimeric G proteins with different alpha subunits, basically. Okay, so we want to follow the activation of these GQ/11 heterotrimeric G proteins. So we need G protein coupled receptors that are capable then of activating heterotrimeric G proteins with these alpha Q/11 subunits as their alpha subunit. Okay, so let me give you some examples of G protein coupled receptors which will bind to and activate GQ/11 heterotrimeric G proteins. So some famous examples are the M3 muscarinic acetylcholine receptor, okay, and also the alpha-1 adrenaline receptor. Okay, so AR for adrenoreceptor or adrenaline receptor. So M3 is a receptor for acetylcholine, which is a G protein coupled receptor. Okay, and alpha-1 adrenoreceptor is a G protein coupled receptor for noradrenaline and also for adrenaline, which I'll just abbreviate down to NA and A respectively. Okay, right, so these are some very notable examples of G protein coupled receptors which, when activated, like to activate GQ/11 heterotrimeric G proteins, i.e. heterotrimeric G proteins where the alpha subunit is either alpha Q or it's alpha 11. Okay, right, so these types of G protein coupled receptors are collectively known as GQ/11 coupled G protein coupled receptors, okay, meaning that when they are activated, they will couple through GQ/11 heterotrimeric G proteins. Okay, right, so let's now say then that we have activated some GQ/11 coupled G protein coupled receptor, okay, with its respective ligands. So we could say it might be the M3 receptor here, and acetylcholine is now bound to that M3 receptor. That's changed the conformation of the M3 receptor so that its intracellular domain has now become available for the binding of GQ/11 heterotrimeric G proteins. The GQ/11 heterotrimeric G protein will then bind to the intracellular domain of the G protein coupled receptor, and then what will happen is it will get activated, okay, and you'll end up with this on-state GQ/11 heterotrimeric G protein. So now what we want to see is what are the signaling effects of this. Now we do not care about the signaling of the beta-gamma complex. What we do care about is what this alpha Q/11 subunit is going to do, okay, because it now is capable of activating phospholipase C enzymes of the beta family. Okay, so let me talk a bit more now about phospholipase C enzymes of that beta family. Okay, so remember, this is the most mainstream family. This is the family of phospholipase C enzymes which can be activated by alpha Q/11 subunits. The other ones, such as phospholipase C gammas, can't. Okay, now, phospholipase C beta enzymes, they move around in the cell. Sometimes they are in the cytoplasm and are soluble, and at other times they are bound to the underside of the phospholipid bilayer. Okay, so let me show you this. So here is a phospholipase C beta enzyme that is floating around in the cytoplasm, and I'll just move this up in a moment. Okay, so here we are. So this is our phospholipase C beta family enzyme, so either phospholipase C beta 1 or beta 2 or beta 3 or beta 4, okay, and they can be floating around in the cytoplasm like this one is at the moment, okay. However, they can also bind to the underside of the phospholipid bilayer, okay, and I want to stress that even when they temporarily bind to the underside of the phospholipid bilayer, they are not activated. At all points here they are inactive. Okay, so here is the phospholipase C beta enzyme. All it's done is relocate, but it's not active. Okay, it's not going to be breaking down PI45P2 molecules yet. Okay, all it's done is changed the place it is located. And of course it can drop off the phospholipid bilayer and go back to the cytoplasm. Okay, so there is this equilibrium basically between phospholipase C beta enzymes, which are moving from being free and soluble in the cytoplasm to being attached to the underside of the phospholipid bilayer, and those which are moving from being attached to the underside of the phospholipid bilayer to being free again in the cytoplasm. Okay, now, the alpha Q/11 subunits of heterotrimeric G proteins are capable of activating those phospholipase C beta enzymes that are currently attached to the underside of the phospholipid bilayer. Okay, so, basically what can now happen is our alpha Q/11 subunit with the GTP bound can bind to these phospholipase C beta enzymes which are attached to the underside of the phospholipid bilayer. So here is our alpha Q/11 subunit here, okay, which I'll colour in once again in red. So it can bind to a phospholipase C beta enzyme whilst it's attached to the underside of the phospholipid bilayer, and now it can result in the activation of this phospholipase C beta enzyme. So now it's activated. Okay, it also results in the stabilization of the attachment between the phospholipase C beta and the plasma membrane. So this isn't about to now just fall off whilst the alpha Q/11 is attached to it. Okay, so it's now active, it's in the right place to catalyze the reaction. Remember, what does it catalyze? The breakdown of PI45P2 into diacylglycerol and IP3. The substrate is in the plasma membrane, so being located underneath the plasma membrane is ideal, basically, so it's in exactly the right place. It's now been activated by the alpha Q/11 subunit with GTP binding to it, and now it's going to start breaking down PI45P2 into diacylglycerol and IP3. So let me show this. So here is PI45P2, and what's now going to happen is it's going to be broken down into diacylglycerol and IP3, which will drop into the cytoplasm. Okay, right. So that is one of the signalling cascades then, a very famous signalling cascade, which can lead to the production of IP3. So it's the cascade that occurs downstream of these GQ/11 coupled G protein coupled receptors. Okay, and we have looked at two important examples of GQ/11 coupled G protein coupled receptors: the M3 the M3 receptor and the alpha-1 adrenoreceptor. Okay, but there are loads more, so loads and loads of extracellular ligands binding to GQ/11 coupled G protein coupled receptors can activate this same cascade within cells and therefore lead to the production of IP3 within the cell, which is going to then act on IP3 receptors and cause a calcium signal within the cell, which we'll come on to later on. Okay, right. So what we now want to turn our attention to is the other family of phospholipase C enzymes that I wanted to talk about, okay, which are activated by very different receptors to the phospholipase C betas. These are activated by these G protein coupled receptor cascades, okay. The phospholipase C gamma enzymes are going to be activated by receptor tyrosine kinases rather than G protein coupled receptor cascades. Okay, so let's now talk then about phospholipase C gamma activation by receptor tyrosine kinases. So firstly, let me talk a little bit about receptor tyrosine kinases like I did for G protein coupled receptors. Okay, so receptor tyrosine kinases, which I previously abbreviated down to RTKs. Okay, now these are basically receptors in the plasma membrane which have an inbuilt tyrosine kinase enzyme, an enzyme that is capable of phosphorylating tyrosine residues, adding phosphate groups onto tyrosine residues. Okay, right, so let's have a look at the structure of receptor tyrosine kinase, but before I just do that, I want to give you the sort of archetypal example of a receptor tyrosine kinase, which you can be thinking of in the back of your head. Okay, so the archetypal example of a receptor tyrosine kinase is the epidermal growth factor receptor, okay, the EGFR. Okay, so this stands for epidermal growth factor, that's the EGF. Okay, and then the R is clearly for receptor. Okay, so I suppose I probably should write out what EGF stands for. So this is the epidermal, that's the E, and then growth factor, that's the GF. Okay, right. So this is a sort of prototypical example of a receptor tyrosine kinase. It's the showcase example. Okay, right. So let me now show you the structure of a receptor tyrosine kinase, which this example, epidermal growth factor receptor, will conform to. Okay, right. So they're in the plasma membrane, just like G protein coupled receptors. So this can represent the plasma membrane here. Okay, and again they are single proteins, so no multi-protein complex here. It's just a single protein. Okay, now the amino terminus will be extracellularly. Okay, so this is the extracellular fluid side of the membrane here. And then you'll have a single membrane spanning alpha helix crossing the membrane. Okay, then you'll have the cytosolic domain, which will contain this extremely important domain, which is the actual tyrosine kinase domain. Okay, so I told you that the characteristic feature that all of these receptor tyrosine kinases share is that they have this inbuilt tyrosine kinase domain here. Okay, and this is what I'm representing in this rather crude way of showing an enzyme. Okay, so this is the tyrosine kinase domain of our receptor tyrosine kinase. Okay, so I will colour in this receptor tyrosine kinase domain in orange. Okay, now, initially then, before the receptor tyrosine kinase has been activated by its ligand, and I should stress that there are loads of different receptor tyrosine kinases, and they'll all have different ligands. The ligand for epidermal growth factor receptor is epidermal growth factor. Okay, but the ligand for different receptor tyrosine kinases will be different. Okay, but they all have this basic structure, with the exception of a few, such as the insulin receptor and the receptors similar to the insulin receptor. Okay, but those are the minority, so we won't go there. Okay, so this is what most receptor tyrosine kinases look like, and this is what the epidermal growth factor receptor looks like. Okay, now, before the receptor tyrosine kinase has been exposed to its ligand then, this tyrosine kinase domain has a very low level of activity. Okay, so it's not really that good at adding phosphate groups onto tyrosine residues. Okay, now, to activate the tyrosine kinase domain of our receptor tyrosine kinase, you have a special tyrosine residue, okay, that needs to be phosphorylated if the receptor tyrosine kinase is to be activated. So if you add a phosphate group onto the tyrosine residue of this tyrosine kinase domain, it will suddenly become much more active, basically. Okay, right. So, let me now say what's going to happen. So, let's go through this cascade. So what's going to happen is the ligand is going to now bind to the extracellular domain of our receptor tyrosine kinase. Okay, so I'll show the ligand here, and I'll colour the ligand in green. So if this is the epidermal growth factor receptor, then this ligand will be the epidermal growth factor then. Okay, what that then triggers is a change in the extracellular domain of the receptor tyrosine kinase, and now what can happen is this receptor tyrosine kinase extracellular domain can dimerize with the extracellular domain of another receptor tyrosine kinase that has ligand bound to it. Okay, so here is another receptor tyrosine kinase coming along that also has got ligand bound to its extracellular domain here, and that's why the two of them can dimerize together like so. Okay, so I want to repeat this, that it is the extracellular domain here that is taking part in this dimerization. Okay, now of course, what this results in is the two receptor tyrosine kinases here, they bring their tyrosine kinase domains very close together. Now, for our purposes, we will simplify this and assume that the two receptor tyrosine kinases here are absolutely identical. Okay, so we will assume that if this one is an epidermal growth factor receptor, this one also is an epidermal growth factor receptor. It's not quite that strict in nature. Okay, they can be different receptor tyrosine kinases, it turns out. Okay, but we won't go there. Okay, we don't need to go there. So we'll just assume that these two receptor tyrosine kinases that are coming together and dimerizing in this way because they have ligand bound to them are identical. Okay, so we've brought our two receptor tyrosine kinases close together. Now, this one's tyrosine kinase domain will also be working at a very low level because it hasn't had this phosphorylation site phosphorylated. Now, here's what happens now. Because you have brought the tyrosine kinase domains so close together because of the dimerization of the extracellular domains of these receptor tyrosine kinases, what ends up happening is the tyrosine kinase domain of one of these, and let's number these up. Let's call this one receptor tyrosine kinase number 1, and let's call this one receptor tyrosine kinase number 2. Basically, the tyrosine kinase domain of receptor tyrosine kinase number 1 is going to end up phosphorylating, eventually, the tyrosine on the tyrosine kinase domain of receptor tyrosine kinase number 2, this activatory phosphorylation site here. Okay? And similarly, the tyrosine kinase domain of receptor tyrosine kinase number 2 will reciprocate and phosphorylate this phosphorylation site of the tyrosine kinase domain of receptor tyrosine kinase number 1. Okay, and we know what will happen now. Okay, both of these tyrosine kinase domains now become much more active, basically. Now they've got that phosphorylation site phosphorylated. Okay, now that step can take a while, okay, because remember their activity is very low, so it's going to take them a while to get around to phosphorylating each other's phosphorylation site like this, but once it's happened, these two now have much higher activity, and what they end up doing is becoming fantastic at phosphorylating tyrosine residues. Okay, and they end up phosphorylating absolutely loads of tyrosine residues in each other's intracellular domain. Okay, so I'm just adding a few more tyrosine residues here. Well, I should have probably said at some point, Y here. Y is the single letter amino acid code for tyrosine. Okay, so that's why I've put this Y here. Okay, these are representing tyrosine residues. Okay, so I should have said that at some point. So Y is the single letter code for tyrosine. Okay, right. Basically, the intracellular domains of these receptor tyrosine kinases will have lots of tyrosines in that could be phosphorylated. And now that these two are activated in this way, they're going to go mad, basically. Each of them is going to start phosphorylating all of the tyrosine residues, or a great chunk of them, in the other's intracellular domain. Okay, so now loads of these tyrosine residues in receptor tyrosine kinase number 2's intracellular domain are going to get phosphorylated, and this phosphorylation is being done by receptor tyrosine kinase number 1 here. Okay, so it's responsible for all of this. Okay, and of course receptor tyrosine kinase number 2's tyrosine kinase domain will reciprocate, it'll do the same to receptor tyrosine kinase number 1. It's going to put loads of phosphate groups onto the tyrosine residues in the intracellular domain of receptor tyrosine kinase number 1, like so. Okay? And this process is known as autophosphorylation, and I will put this word over here, okay, because I want to leave this space free. Okay, so this is called autophosphorylation. Okay, now auto means self. Okay, so this means self-phosphorylation, and you might be wondering, well, why is that self-phosphorylation? Okay, because it's different receptor tyrosine kinases phosphorylating the other one, basically. Okay, but if you view the dimer as being one structure in its own right, and there is good reason for viewing it as one structure in its own right, because in the case of the insulin receptors and receptors similar to the insulin receptor, the receptor is pre-dimerized long before the ligand ever arrives. Okay, so because the insulin receptor is so heavily studied, and it's the origin of most of our knowledge about receptor tyrosine kinases, okay, that's why this terminology came about, that it's autophosphorylation, and why you view the dimer as being one entity on its own. Okay, right. You now then have your activated receptor tyrosine kinase dimer here. Okay, it's the appearance of all of these phosphorylated tyrosine residues that is now going to activate intracellular signaling cascades. Now, receptor tyrosine kinase dimers like this can activate a huge number of different intracellular signaling cascades. So these phosphorylated tyrosine residues, and I'll give you a piece of terminology here. So the way that you can, the name for a phosphorylated tyrosine residue is a phosphotyrosine residue. Okay, so now we've got the appearance of all of these phosphotyrosine residues on the intracellular aspect of the receptor tyrosine kinase dimer here. Loads of different signaling proteins can come and bind to these phosphotyrosine residues and then set up all sorts of different intracellular signaling cascades, basically. Okay, now, we're not going to go through all of the different signaling cascades that receptor tyrosine kinases can activate. The one that we're going to be interested in is the activation of phospholipase C gamma enzymes. So, let me now show you this. So basically, phospholipase C gamma enzymes have a special domain that allows them to bind to phosphotyrosine residues. So this is my picture of a phospholipase C gamma enzyme. The entire thing is a phospholipase C gamma enzyme. Okay, and it has this special domain here which is binding to a phosphotyrosine residue. Okay, and this is a domain that you find in many different proteins that are activated by receptor tyrosine kinases, and they all use this domain to bind to these phosphotyrosine residues. Okay, and this domain is what's known as an SH2 domain. Okay, and it stands for Sark homology 2 domain. So like EF hand domains, Sark homology 2 domains are a domain that you can find in lots of different proteins that all look very similar. So if you look at these different domains and different proteins, they all look similar enough to be counted as the same sort of domain, basically. Okay, and this type of domain was first found in a protein called SARC, basically. So this is the SARC homology domain 2. So SARC is a non-receptor tyrosine kinase. Okay, it's a free receptor tyrosine kinase. It's a free tyrosine kinase, okay? So it's not attached to a portion that is an integral membrane portion, okay? It's just free and floating around in the cytoplasm. Okay, so SARC has one of these SH2 domains, basically, and it was one of the proteins that this domain was first found in, hence why it's now called SARC homology domain, the domain that is homologous to a domain that we have found in SARC. Okay, so SARC homology domain 2s, or SH2 domains, are often used by proteins to allow that protein to bind to phosphotyrosine residues, particularly on the intracellular aspect of receptor tyrosine kinases, as is the case here for the phospholipase C gamma 2 enzyme. Okay, so previously, before the phospholipase C gamma 2 enzyme had bound to the phosphotyrosine residue like so, it was floating around in the cytoplasm. Okay, it's not like phospholipase C beta. It cannot bind to the underside of the phospholipid bilayer on its own. Okay, so remember, phospholipase C beta could move between floating around in the cytoplasm and being attached to the underside of the phospholipid bilayer. Phospholipase C gamma isn't that skilled. It cannot do that on its own. It can only float around in the cytoplasm. But when you get the activation of a receptor tyrosine kinase dimer like this, it can attach to the phosphotyrosine residues on the intracellular aspect of that receptor tyrosine kinase dimer, and hence it can relocate itself nicely close to the underside of the phospholipid bilayer, which is awfully convenient because that's where its substrate is. That's where the PI45B2 is. Okay, so it's now located itself nice and close to where its substrate is. However, that doesn't activate it, that just locates it in the right place. What then activates the phospholipase C gamma enzymes is that they end up getting phosphorylated by one of these tyrosine kinase domains of the receptor tyrosine kinase dimer. Okay, so one of these tyrosine kinase domains is then going to phosphorylate a tyrosine residue within the phospholipase C gamma. In fact, they phosphorylate many different tyrosine residues within the phospholipase C gamma, but there is one tyrosine residue that is particularly important. Again, it's the phosphorylation of this one that causes the phospholipase C gamma enzyme to then activate. Okay? So now, once it's been phosphorylated like that, it's now going to be active. And then, of course, what it's going to do is it's going to find its substrate in the underside of the phospholipid bilayer, which is the PI45B2, okay, and it's then going to break the PI45B2 down into diacylglycerol and IP3. Diacylglycerol will remain in the phospholipid bilayer, and the IP3 will drop into the cytoplasm and then activate the IP3 receptors, leading to a calcium signal. So the exact same signaling pathway can be initiated by receptor tyrosine kinases as it can through G protein coupled receptors, through these different family of phospholipase C enzymes. Okay, so phospholipase C beta enzymes are the most famous ones. They're part of this classical G protein cascade, but phospholipase C gammas are notable as well. They act downstream of receptor tyrosine kinases. Okay, so we'll call it there for this video, and in the next video we will now discuss what IP3 is actually going to do, how it's going to work on IP3 receptors. We've discussed its generation now we'll move on to what it's actually going to do.